Hello, this is Mr. Laudermouth. Good morning, good afternoon, or good evening to you, as the case may be. This is part 2 of my screencast on meiosis and sexual reproduction. When we concluded the last video, we had just gone through interphase prior to starting meiosis. So all of our chromosomes in the nucleus now had sister chromatids. They were double-stranded chromosomes. So we are now ready to begin meiosis 1. First of all, we got to realize the goal of meiosis 1 is to separate homologous pairs of chromosomes, pairs that contain the same genes and are the same size. Each of these homologous pairs will form what's called a tetrad. Basically, they will sort of line up next to each other and overlap in prophase 1. That overlap is called a synapsis. When they're finished with prophase 1, each pair lines up along the midline of the cell. They line up along the equator in metaphase 1. The pairs then separate in anaphase 1, and in telophase 1, we now have separate nuclei, each with just half the number of chromosomes we had at the beginning. So this is our reduction division, taking us down to half the normal number of chromosomes, to haploid cells. So we've successfully separated homologous pairs in meiosis 1. Okay. Meiosis 2. This is our second division in the meiosis process, and this will separate sister chromatids. Now, if you remember back to mitosis, we were separating sister chromatids in mitosis as well. So these steps in meiosis 2 are quite similar to mitosis, but we got to remember the important difference. Here we've already got haploid cells. So we'll go through prophase 2, metaphase 2, where each chromosome lines up along the equator. Anaphase 2, we separate the sister chromatids. They move to separate ends of the cell. And in telophase 2, we get separate nuclei, each of which is still haploid. Notice how at the start of prophase 2, each cell had 2 chromosomes. At the end of at the end of meiosis 2, we have 2 chromosomes in each cell, and they are not identical. If you look and compare these 4 haploid daughter cells, they don't have exactly the same chromosomes. They're from the same homologous pairs, but they contain some difference in genes, very likely, differences in alleles. Okay, so this is the process that, for instance, could get us to 4 sperm cells, 4 male gametes. So to kind of do a quick overview of all the steps in meiosis, we start with interphase, where we copy our DNA, where we make those double-stranded chromosomes. We then divide up our homologous pairs. We do our reduction division, going from prophase 1 through telophase 1. Meiosis 2, our second division, separates the sister chromatids. This time we're going from haploid to haploid. It's very similar steps to mitosis, with the exception that we are not making identical cells, and the cells are haploid, not diploid. So there are pictures of those steps in an animal cell. We can see prophase 1, the homologous pairs, into metaphase, anaphase, and then metaphase, anaphase, telophase 2, getting us to 4 haploid cells. So during prophase 1, I mentioned that the homologous pairs of chromosomes will overlap when they form that tetrad structure. They specifically are doing that so that they can swap sections of DNA. It's a process called crossing over. When they intertwine, they can exchange genes. They can actually exchange segments of DNA. As we see in this picture, the red and the blue chromosomes have swapped some of their DNA. They've traded some of their genes. Literally, this involves breaking and reattaching pieces of the chromosomes. So we have the crossing over, the breaking of the DNA, the refusing of the DNA. That gets us now what we call recombinant chromosomes. They've been recombined. Each chromosome now contains genes that it did not have before. Is there an advantage to crossing over in terms of sexual reproduction? Well, the answer is yes, there is. Because by recombining genes through crossing over, we can have offspring with new combinations of traits. Some of those combinations will result in phenotypes that are better suited for the environment. Those individuals will have a reproductive advantage. Natural selection will favor them, and that could actually cause a shift in traits in the entire population over time. So this, again, is a source of variation that natural selection can work on. It's one of the reasons why sexual reproduction works so well hand in hand with evolution. It's providing the fuel, if you will, if it's providing the variation for natural selection to work on. So this chart kind of nicely summarizes differences between mitosis and meiosis. Mitosis, in prophase, we do not have crossing over. We do in prophase 1 of meiosis. In metaphase of mitosis, each chromosome lines up along the equator, and we split the sister chromatids. In metaphase 1, it's the pairs, the tetrads, that line up and separate during during anaphase 1. The sister chromatids don't separate in meiosis until we get to meiosis 2, to anaphase 2. At the very end of mitosis, we will have two daughter cells that are identical to each other and just like the parent cell we started with. In meiosis, we're producing 4 daughter cells that are haploid. Notice none of them are exactly like their neighbors, and they're certainly not like the parent we started with. The parent cell up here had 4 chromosomes. Each one of these only has 2. So 1 division versus 2 divisions, identical cells versus genetically different cells, 2 cells versus 4 cells, diploid to diploid, diploid to haploid, producing cells for growth and repair, producing cells for reproduction, gametes. No crossing over in mitosis, but definitely crossing over in meiosis. So this is a good slide to know the differences between mitosis and meiosis, a topic that comes up fairly frequently on the AP exam. When we put all our knowledge together and we take the process of meiosis in females, this will get us to an egg cell with 23 chromosomes, and males, sperm cells with 23 chromosomes. Those gametes can combine in the process of fertilization, creating a zygote, a new organism that is back to the 46 chromosome number, back to the diploid number. That zygote can then begin dividing by mitosis. Each new cell of mitosis will also have 46, and it's that process that gets us from a single-celled zygote to a multi-celled human being with trillions of cells. Okay. So mitosis leads into embryonic development. Okay, to recap here, what's the value of sexual reproduction to multi-celled organisms? Well, it introduces genetic variation in a couple different ways. There's genetic recombination that happens when the chromosomes separate from each other during meiosis to form gametes. Notice in this particular picture down here, we don't have to line up the homologous pairs the same way every time. So simply by switching which chromosome is on which side, we guarantee that we're going to produce gametes that are genetically different from each other. There's a random alignment during metaphase 1. Crossing over back in prophase 1, we know that's going to mix up some alleles. It's going to mix up some genes because the homologous pairs will swap. It's going to create chromosomes that did not exist before in the parent organism. And of course, fertilization is a random process. Now in humans, we don't generally think of picking our mates as a random process, but certainly which sperm cell combines with which egg is a largely random process. And since we don't know ahead of time what combination of genes are in those gametes, it's going to result in a zygote with basically a randomized collection of chromosomes and genes. All of this genetic recombination, crossing over, and random fertilization combine to create individuals within a population who show variation in their traits, like this population of beetles. They're all members of the same species, but clearly they show a difference in traits because of these factors. In evolution, this variation is what natural selection will be picking, deciding which combination of traits is better suited for a particular environment. Those individuals will survive and have a greater chance of passing on their combination, or at least some of their combination of traits, to their offspring. So, if we have independent assortment that's introducing genetic variation during meiosis, we have gametes in the offspring that do not have to have the same combination of genes as those in the parents. So mom and dad's genetic material combined to create the offspring. When the offspring does meiosis to make gametes again, because of the factors we just discussed, we expect that the gametes in the offspring will have different combinations of genes than what was originally in the parents. So children and their parents do not have to be genetic clones of each other. This random assortment, by the way, it just by itself means that if all we're counting is chromosome pairs here, there's 2 to the 23 different ways we can combine chromosomes to produce gametes. That's over 8 million combinations before we even factor in crossing over or the random combination of egg and sperm. So this process just by itself, the random assortment that happens during meiosis, is going to create 8 million different combinations. Okay. This variation that happens in crossing over, of course, introduces nearly an infinite variety of differences in gametes because there's no set formula in crossing over that says these 2 particular pieces of DNA have to swap. Certain pieces of DNA will swap in some homologous chromosome pairs and not others. And it seems to be a largely random process in each particular case of meiosis, in each particular prophase 1 event. Because the tetrads are all independent of each other, we can get almost an infinite variety of crossing over, producing an almost infinite variety of genetically recombined chromosomes. And of course, random fertilization. Any 2 parents can produce a zygote with over 70 trillion possible diploid combinations. If we take that 2 to the 23rd power that we talked about from our random assortment of chromosomes, and we factor in the fact that each individual had 2 parents, that's 2 to the 23rd squared, that ends up over 70 trillion combinations before we factor in crossing over. So another way to think about that, even if crossing over never happened, 2 parents, a particular mother and father, could have over 70 trillion children before any one of them would have to be genetically identical to any other. Okay. So we know that sexual reproduction results in variability. It allows a population that is, while able to interbreed, it's the same species, to still show variation in its traits. So we can see that within a family. If we look at the Baldwins here, clearly they show some traits in common. We can sort of recognize right away that they're members of a genetically related group, and yet we can also distinguish them from each other. They show variation in some traits and not in others. The Jonas brothers, much the same way. The Sheen-Estevez family, much the same way. We can note genetic similarities, but also genetic differences as the result of this variation that comes from assortment of chromosomes, crossing over, and random fertilization. So we kind of want to wrap up this video by just talking about the difference between sperm production in men versus egg production in women, because there is an important difference there. Sperm production follows the pattern of meiosis we've been studying. There's a meiosis 1, a meiosis 2, and at the end you get 4 haploid, non-identical sperm cells. This process basically begins when a male reaches puberty and more or less continues for the rest of that individual's life, resulting in millions of different sperm. Each one is genetically different, essentially, from the rest. Egg production, which is known as oogenesis, this actually occurs in stages. It's not a continuous process. The eggs in the ovaries will actually stop developing at anaphase 1. Once a woman reaches puberty, once a month during the ovulation cycle, one of those egg cells, or I should say kind of a proto-egg cell that was stuck in meiosis 1 is going to continue through the other stages of meiosis, and in fact won't even finish all of meiosis unless it gets fertilized by a sperm. Along the way, during these cell divisions, there's an unequal dividing of the cell. Normally, we would think of cell division producing two cells with equal amounts of cytoplasm. That doesn't happen in oogenesis. Instead, we get one cell that's got virtually all of the cytoplasm, mitochondria, ER, ribosomes, almost all the cell parts and cytoplasm in one cell. This other little cell that's called a polar body, all it is a waste receptacle for the chromosomes we're trying to get rid of. Remember that we want to produce a cell that's haploid, so we have to lose half the chromosomes along the way. That's what this polar body is helping us do. But what we don't want to lose along the way are any cytoplasm or other cell organelles. This happens again during meiosis 2. There's a division, and we get rid of the unwanted chromosomes with this other polar body, and we keep the egg cell by itself with its chromosomes and all the cytoplasm and other organelles. Why do it this way? Well, it's because the sperm cell can only carry chromosomes. It has to travel a long distance to basically avoid being weighted down. The only thing that the sperm cell really carries with it is its share of the chromosomes. It's 23 chromosomes. That means all the other cell parts that the zygote is going to need, all the ER, the mitochondria, the ribosomes, all those other cell organelles have to come from the egg cell. They have to come from the mother. So it's kind of interesting here. If we're talking about your genes, your chromosomes, you did get half from mom and half from dad. But if we're talking about your mitochondria or your ER or your lysosomes or your Golgi, you actually inherited all of that from your mother, none from your father. So there's oogenesis as we see it's happening. Again, along the way, we're losing these polar bodies. They're a way of getting rid of unwanted chromosomes so that in the end we get just one haploid egg cell. So oogenesis, at the end, we get only one functional gamete. Spermatogenesis, we got 4. Okay. Are there differences in meiosis across the kingdoms? There are some subtle differences. For instance, in some organisms, we'll find that it's actually the haploid stage of the life cycle that's the more dominant. In some types of algae, for instance, most of the time the organism is in a haploid state, not a diploid state. In other organisms, it's more evenly divided. For instance, in these ferns, they spend about half their life cycle as a haploid individual, a totally separate plant, and about half their life cycle as a diploid individual, the big fern with the fronds that we're used to thinking of. Most animals, they're spending pretty much their whole life cycle as a diploid individual. The haploid stage doesn't last that long. So there can be an alternation here between haploid and diploid that lasts a greater or longer period of time. But if you're going to do sexual reproduction at some stage, you have to go from haploid to diploid to haploid to diploid. There has to be a trading off. All right, I hope this video was helpful, and I'll see you in class.